Okay, welcome back. So Alex was reminding me that last year when he gave this talk, he was not yet in post in Manchester, and he was actually in Argentina when he gave the talk. So this year we're happy to have him in person in the UK, in Manchester even, which is amazing. So Alex is the bicentennial Turing Chair of Computational Medicine, and he started in Manchester about a year ago, and he's director of the Pankhurst Institute for Health Technology Innovation or something, I think. and is working very hard to kind of develop the vision of the Pankhurst so that people like me actually can describe it in a smaller number of words, yes. And is really at the forefront of developments in In-Silico trials, which is a really exciting area, which involves sort of dealing with regulatory aspects. And Alex has really been working hard to get these things, you know, through regulatory hurdles and things. And I think he's going to tell us something about that today, probably.
Yeah.
So, over to you, Alex.
Thanks a lot, Magnus. And it's great to be here. So I was a bit, had a lot of thought about what to present today, especially what would be the angle that you would be most interested, and also how to be honest to myself in terms of what I believe is the best thing that I can give to you, right? And I came to something which I hope you will like. I hope it strikes the right balance between what I believe is that to smart people, and you're all smart people, you don't give them details of how to solve problems. You tell them the biggest questions that you can think about and how to think about questions themselves, because they will figure out solutions. And at the same time, you know, feeling that if I was to do only that, you may get a sense that it was very high level and you wouldn't know really what I'm doing. So I try to pepper in a few areas some concrete methods that we have developed to give you that view. And then I did the third thing, which is last week there was a conference at the Isaac Newton Institute of Mathematical Science in Cambridge, where a bunch of people across the UK that work on cardiac modeling met together. And there was a lady in the audience that kept asking to every single speaker, can you say something that is useful for early careers as well? So I came up with some thoughts that just to prevent that someone would ask those questions, I put some answers to that as well. So first, what is the Christopher Pankhurst Institute that Magnus was talking about? So this is basically a partnership between the university, the three faculties, and then a number of other actors in the city, including Manchester Foundation Trust, hospitals, so Manchester Infirmary and other hospitals from the same group, and also the region government. We're in the process of finalizing the strategy, but what you see there is a bit of a summary, so it's a bit of a preview for some of you. But we're going to be focusing, essentially what we want to do is provide leadership to health technology in the broader sense by pioneering challenge-driven discoveries that lead to patient benefit and economic growth. So the metrics of success for the Institute is how many companies we generate, how many new products are in the NHS as a consequence of providing, you know, solid scientific discoveries that actually help to drive unmet health and care needs. Now, this is massive, so what exactly where we do that? We do that primarily in the areas of AI and digital health, in computational medicine and biology, in advanced interventions and devices. And we take opportunities that we think are happening at the moment, which is there's a huge of technological convergence, so software and hardware and chemistry all coming together, and the previous talk shows some of those convergences between, say, chemistry and AI. Also the importance of multimodal data integration, and then the importance of regulatory science, which may sound to you a lot to paperwork and boring stuff and something that is done in humanities, not here sorry, not that humanities do boring stuff, but what I mean is that there are law and these sort of things. But actually there's a lot of engineering to be made and a lot of mathematics and a lot of, you know, solid computer science that needs to happen. And then because we are, you know, challenge driven, there is 4 challenges where we are working on. These are not technology driven, these are needs driven, which are in population health, in early diagnosis, in developing better interventions faster and sustainably, and also in how we put all of these under an ecosystem that is digital within the city. Next year, we're organizing, if you're interested in what I'm going to talk today, next year in June, about this time, there will be the second time this conference happens, but the first time it's going to happen in Europe on virtual imaging trials. So I'm going to talk today mostly about medical devices, but mainly therapeutic devices. But this is essentially in-silico trials for diagnostic devices and also to how you do biomarker discovery as well through modeling and simulation. Now, if you get very confused at the end of this talk, you go back to this slide. I will tell you how I put it all together in my head, okay? So the big question that I'm trying to answer is how I combine data-driven with mechanistic models. The first ones are limited by the amount of data, while the seconds are limited by the knowledge we have from basic science. The first one is limited by the sampling abilities that we have, while the second one is limited by the theory that we have about each of them. Now, you may ask why this is important, because if you look over the next, say, 10 years, how the amount of demands in complexity, data volume, processing power will grow, according to a few people who are doing, so I'm a chair in computational medicine. If you think that's crazy, the guy who wrote this paper is a chair in computational sociology, okay, which is even more abstract to me. But he's a physicist, a theoretical physicist himself. And I'm an engineer myself. And what he developed is a model that says, look, over the next few years, processing power will continue to grow according to the Moore's law, so doubling every 12 months, every 18 months. Data is currently doubling every 12 months, but it's also exponential. And there are people that say that in the not-too-distant future, it will double every 12 hours. But nonetheless, it's exponential, and that's the important thing. When you have two exponentials and one is faster than the other one, they cross at a given point. Right? This means that fairly soon we will have more data that we will be able to compute. That's a problem. And then the other problem is that the complexity of the systems we are studying, whether it is a neural network with the increasing number of parameters or it is a biological system with our ever growing understanding of what's happening, it's growing factorially as opposed to exponentially, which means that much faster than these other two, there will be a point that the complexity of the systems we are trying to identify through parameter identification is much bigger than any amount of data we will be able to collect and much bigger than we will ever be able to compute. So what I'm telling you with these data-driven methods suck, because at some point you won't be able to understand the system just with data-driven models. And that's why I believe you need to have mechanisms as well, because mechanisms allows you to shortcut this course of scale by incorporating priors. Okay? And this shows you a bit the available data that we have and the amount of mechanisms that we understand. So we cannot have, if we had only first principles and we were able to understand everything, we didn't need data-driven models. If we had lots of data, we don't need to understand anything because we could potentially create models from the data. But the reality is we are in neither of these two extremes. And we are always in some middle ground where we have some level of understanding and some amounts of data. And if I go to my previous speaker, she was saying that one of the biggest questions is how you sort out this problem of data sparsity. And that's potentially one way to think about solutions to that problem. Okay. So if you are familiar with the DAKY pyramid, so the data, information, knowledge, and wisdom, so the question is we have lots of data, we actually need wisdom, so guidelines, principles, so that we can take decisions, because actions come from wisdom, not from data. Data in itself is not enough. And the way to go from data to wisdom is by information processing and knowledge modeling, which is what we're going to be talking all the time. And therefore you have phenomenological models that go bottom up, and mechanistic models that go top-down. And the problem is that sometimes you don't have either of the two extremes, and you need to go middle out. So you need to go from some level of understanding of the models and the basic principles, so you have some epistemic understanding or knowledge, but also uncertainty, and also you have measurement uncertainty and aleatoric uncertainty. And those 2 points need to sort of meet together. This is just a point that maybe we can come at the end, but I tend to think many people have a big problem about figuring out whether they are theoreticians or applied. I don't care. What I believe is that we need to be mission-driven, that we need to have a clear problem that we're trying to solve and be smart enough to know when we're applying techniques and when we need to develop completely new techniques. And I think the next generation of smart people will be those that understand that fully, in the sense that are what I call translational scientists, not translation in the sense to medicine only, but translation in the sense that they translate across disciplines and domains. And there's a number of characteristics that I'm not going to go through those, but if you're interested, there's a nice paper that actually explains a bit about this. Now, what is my mission then? Well, my mission is the one that clinicians have, in a way, which is the first principle of not harming. Okay? Now, the way I understand not harming is about how I help develop methods that enable to develop technologies that reduce the amount of harm. So let's think, as I explained in my question earlier, I don't do drugs in the sense of do any research in that space, but I do understand a bit what they do there. But whether you are in a drug cycle or in a medical device cycle, which is where I focus, it's a lengthy process. 15 years, you heard it before, 1.1 billion to develop a drug. You'll hear in a minute the same numbers, but for devices. This is probably 15 years. This is about 11 years, a bit shorter, but it's still very lengthy. Now, all of the whole theory of how you develop new technologies is predicated on three sources of scientific evidence. So when we talk about regulatory evidence, we're actually talking about scientific evidence. So if you know how to do science, you know how to do regulatory evidence. And there are three ways: bench testing, animal testing, and human testing. And what are the questions? You are asking yourself questions about safety. Is my product, on the basis of the evidence that I have, safer, will do more good than harm? And efficacy, does it do any benefit to the patient? You can do no harm and no benefit, right? Now, the question is does it work, this system? And I hope to convince you that this actually also sucks and it doesn't work. So why? Because, again, I'm not going to go in the detail of all of the limits of each technique, but bench testing has a number of issues that you can read there. Animal testing, the same. Most of the animal models, the results from animals don't translate to humans. And doing clinical trials, some people think is the holy grail, but actually is very difficult as well. Is full of issues like biases, potentially ethical and logistical considerations, costs which are enormous, and you're putting people at risk, at least during the trial. And the other thing is you can do all of the things wrongly. This is called bad science, and of course there is always a lot of that as well. But even if you do it perfect, there are issues behind. The other thing is, which of these ones you think is a ground truth? So if you know which one of those is the ground truth. Which of these is a model? What is it? Why? Explain that. That's absolutely right. Let me ask you a question. Someone asked me once, because I made that point and said, no, human trials are not a model. That's the fact. Why is not true that?
Because the ground truth is using them everyday people all the time.
20%, right?
Yeah.
And a trial, a human trial?
Possibly self-select group of people who it's like with lots of things where there's so many carve-outs that disqualify someone from being in a trial.
Is that called inclusion and exclusion criteria? Yeah.
Yeah. Then you only have it's not a model of the system. It's a really it's a model of the system where whatever you're developing has to be applied.
Yeah. So humans, as much as the other ones, of course, they are models of what will happen on the general population. And you do that on a cohort, on a subset that is limited in size, in selection criteria, in potential bias, in experimental limitations, right? So all of them are models. So if anybody tells you a randomized controlled trial is a ground truth, that's not true. It's a model. It's perhaps the best model we have, the most useful model we have, but it's a model nonetheless. Yeah? Okay. So this is a recent paper that looks at complex, meaning not bandages and syringes, okay? So stents, heart valves, pacemakers, complex therapeutic devices. Average cost, 54 million. but it has a massive range for developing. And this is without capitalization cost. This is if you knew from day one the right way to do it, and you spend just the money to get it right, to get it done. But actually, you do lots of things wrong on the way, because actually you don't know what is the right thing. And if you add those capitalization costs, it's 10 times more expensive. Okay? So an expensive medical device can be as bad as a drug in developing it. And then if you look at these numbers, these are what I call the attrition rates, or the death rates of, depending on in which phase you are, how many of those don't get to the next level. So by the time you get here, you have only about 10% of the funnel of innovations you started with. Okay? So who is paying all of that? All of this failure? All of us. The NHS. That's why a device that is a bunch of wire costs £10,000. How can they cost so much? It's not the device what costs, it's everything that you had to do to get to that little bunch of wire. Now, look at this. This is, if you look at, in about 200 studies, trials, randomized controlled trials, they look at FDA determined root causes of recalls. Recall is when there is a failure of a device and you need to take them out of the market, right? Class 1 devices are the most dangerous ones, so the invasive, sort of implanted type of devices. The 43 of those were Class 1 recalls. 53% of the recalls, device design problems. So we don't know how to design devices. 30% of the 200 cases of all the recalls. This is Class 1 on any class. Implant file. 10 years worth of data on a number of devices, 83,000 deaths, 1.7 million serious adverse effects in the US. If you think that that's the US not getting it right, look at these numbers for the UK. So this is what is called the Cumberledge report that look at both drugs and devices in the UK. Is heartbreaking to listen there in YouTube. All the evidence from all the patients that gave to the panel, they're edited so that they took certain things out, names of people and so on, but you can hear them, and lives that were really seriously affected. This is a recall for about 4 weeks ago. This is the second time that a left ventricular assist device, so this is a prosthetic heart valve, gets taken out of the market. 2 years ago, there was another one from Medtronic, and I don't think there is any LVAD anymore out of the market, because the ones that were there, they all failed. Recalled, this is an Abbott device, 273 injuries, 14 deaths. This list here is just the recalls from 2024. Okay? So we're doing great, right? With this approach that we have. So for me, first do not harm is the same than simulation first. And I'll try to explain what that means. So we need to think, I think, differently. Because there are other sectors, like the aerospace, automotive, we will not be tolerating that level of failure that we have in medical devices. And where, if you look at those, they're doing already virtual tests for a number of years, and they're moving into that direction. Now, you may say, yeah, but that's easy. That's not healthcare. Come on. This is highly regulated, mission-critical, multi-scale systems, huge amount of uncertainty, all the same fundamental problems that you have in medicine. So what is an in-silico trial, and what is computational medicine, for the matter? So computational medicine, or in-silico medicine, is applying engineering, math, computer science to improve understanding of disease and improvement of therapies and treatment. An in-silico test, if you are thinking about preclinical or in-silico trials, if you're thinking about clinicals, are computer-based trials using detailed prediction models. And by detail, what I mean is mechanistic, not just phenomenological, in highly controlled virtual conditions or virtual cohorts that represent realistic operational conditions or target populations. So let me explain you in this way. So this is the life cycle of the product in the horizontal. And the vertical one is the number of problems that you need to identify and resolve in the design of a product. What we do at the moment is this line here. So someone, a clinician or an engineer, has a crazy good idea about a new device that you can come up that will benefit patients. They create some initial prototype, they do a bit of testing, and then they're desperate that they need cash to actually do the rest of the research. They go to some investors and they say, this is our idea, we need, I don't know, 2 million quid of a seed capital or 3 million. Eventually they convince the investors, and then they will put a huge amount of demand on them to demonstrate the evidence so that the next round they can inject another 20, 30 million. So the pressure in the system makes them not spend lots of time testing and going as quick as possible to prototyping and preclinical testing, because they don't have cash to burn at that stage. So what you do is you constrain your design space very early on to the space of the possible of being testable and testing. And you do that on bench and you do on animals. Then eventually you get your next round and you start doing the clinical testing and the regulatory approval. And everything that could have gone wrong starts going wrong. Therefore, you have a larger number of problems that you need to solve. The problem is that some of them, well, all of them, at that stage, fixing them, the cost of fixing a unit problem is about 10 to 100 times higher than it was before when you were at early phases. Among other things, because depending on what type of mistake you do, that can have a risk. Especially, there's another box here, which is the one for the later ranges. This is if you were lucky, between quotes, that you passed through here and you went to clinical trials, and then you start finding the problems, right? So the whole idea of in-silico methods is, can you push this envelope earlier on so that through virtual means you can do more inexpensive, more thorough, more diverse set of experiments, so that before you get to this stage you have highly de-risked your innovation, so that you only go with things that have high chances of success. You may still need to do human testing and animal testing and all of that, but you do that as a confirmatory of your design, not as the way to figure out whether you have some legs to carry on running. So for instance, if I show you, I have a slide that I'm not showing today, that I show 1990 a Citroën from that time, and then 2020 and a Tesla, and you would imagine, even if I don't have this slide, there has been a massive change in the product, right, in the automotive industry. If I show you 30 years worth of innovations in brain aneurysms, which is therapy, which is something I'm going to talk a bit later, you will see that it's still a bunch of wires. And we have a still 20% complication rate, 3% mortality. We can do the same type of simulations we do in aerospace. So the question is, if you have 4 ways to find scientific evidence, modeling and the other 3, why would you use only 3? And which one is good? Well, all of them are wrong, fundamentally, but some of them can be useful in different stages. So it's a completely different way about thinking about scientific evidence and scientific regulatory evidence. And my view, and the view of many other people, is that in the future we will use all sources of evidence, selecting them appropriately in a sort of a risk-informed decision-making and credibility framework. And that's why ultimately this is highly connected with our CDT and decision-making, and how actually bring all of these different sources together. So what can you use these in-silico methods for? You can use them to refine trials. For instance, if you have an idea about the magnitude of the effect you're trying to measure, you might be able to do better powering of the designs. You can use it for reducing the number of experiments. So for instance, if I have 10 different innovations for the same prescription, and I could figure out through modeling and simulation that half of them will never make it, or they are the worst that you can think about the 10 that you have at hand, you might be able to avoid 5 trials that otherwise will require a lot of expense and a lot of people being put at risk. And eventually you can replace. I don't think on the short term we are going to replace randomized controlled trials, but I think we will replace voids or regulatory evidence. For instance, in areas where there is simply no regulatory randomized controlled trial, pediatrics, rare diseases, women who are pregnant, which normally are excluded of pretty much every trial for good reasons, but actually end up being bad reasons because they also get ill and you need to treat them somehow. Most of the evidence about drugs and devices for children are actually inferences we make from what happens to adults, but there aren't proper trials in most cases. Okay. Now, is this in prime time? Well, there is a lot of work going on internationally from certain standards organization in the US, and then more recently, FDA has produced last year, look at the dates, July, November, September. This is 2016, but these 2 are actually last year. This is how you do qualification of simulation tools and how you do credibility assessment for computational modeling in medical device submissions. If you look at that, and remember, this is a machine learning bunch, it's all about uncertainty quantification, risk assessment. It's about understanding the propagation or error through those models, because these are complex systems. And there is also communities that are developing good simulation practices and how we should do things properly. And I think this community needs to be involved in those. So a year ago, we did a we published a landscape report, and within that landscape report there was a survey that we did 2 years ago on barriers and enablers for this. And the top 3 of them are CMS stands for Computational Modeling and Simulation and In-Silico trials. So there is uncertainty on the regulatory acceptance of this evidence. There is insufficient confidence on the maturity and credibility of these models. So this is all about model verification and validation. And there is also insufficient expertise and skills in the regulators. They already have issues with AI. Imagine with finite elements models and more sophisticated techniques. So a few weeks ago, with someone from MHRA, we gave a seminar in an organization called NIFEMS, that is a trade organization or membership organization of all of the simulation companies. And they say, okay, you tell that you're uncertain about what we believe. Well, these are the biggest barriers for this is what we think is the right way to go forward. Use the modeling and simulation evidence to plug evidence gap, optimize trial design, reduce the need for unnecessary exposure to an untested intervention on product with unclear benefit, reduce the need for animal testing. This is where they recommend this might work. It's premature to replace fully traditional clinical trials. Focus, however, in enhancing the type of evidence that regulators currently receive and how evidence gaps can be filled. For instance, pediatrics, rare diseases, combination devices, and I'll show an example later. Situations where a randomized controlled trial is a combinatorial design, and you simply wouldn't have the money in the world to do that. And then things like assurance about the quality and completeness of the data that is used to inform the model parameters. So this also is automatic data quality control. These are machine learning problems as well. Can you develop techniques that tell you whether the quality of the data is good or not? The assumptions of the models need to be grounded in science, of course. The models need to be validated. It's all about validation and verification of models. Accuracy of any algorithms used should be verifiable. So how we do the experiments matters. Having good guidelines is important. And then in the context of in-silico trials, using virtual patient cohorts and in-silico models should demonstrate fidelity to the real patient data counterparts or comparative performance versus models using real data. Okay? So all of these are methodology questions that require this community to develop. Okay, so if an in-silico trial was a cake, these are the 3 ingredients of the cake. Okay? The first one is how you build virtual patients and populations. The second one is how you build virtual examinations, if it's diagnostics or interventions, if it's therapeutic. And then finally, how you do virtual readers or readouts. Because you will see that with modeling and simulation, we are unlikely to predict death or pain or things which normally we use, or quality of life, which we normally measure in a trial. But we are actually able to predict a lot of what we call technical outcomes, things like reduction in pressure, increase in leakage through valves. So things that are technical and therefore are closer to the root cause of the success or the failure of those devices. But we might want to connect those with the clinical readouts that clinicians normally are interested in. So how this maps into AI and machine learning, and I'll give some examples later, but virtual patients and virtual populations is all about generalizations of synthetic data, developing of anatomical models and variability and generative AI, develop multimodality, essentially models, how to incorporate trajectories due to aging, lifestyle, comorbidities, complex covariates, so how you may build complex models. Virtual examinations and interventions is all about incorporating physics, biology, physiology. So this is about first principles and how you accelerate potentially them through machine learning and AI. How you incorporate uncertainty in them. How you incorporate multi-scale couplings into those models. And then I'll leave you the final one. So I'm going to skip this one. It will come, I think I have that duplicated, sorry. So let me show you some examples of putting this all together, because I guess so far it's a bit theoretical, all of this, or abstract. And there are, in this landscape report that we published last year, there is about 12 case studies. I'm going to pick one of them and 2 others from my own research. But if you're interested, you can have a look. There is a number of organ systems and in both devices and drugs that you can look at. So all the work that I do, I'm focusing on two driving exemplars, because I need myself to focus on something to be able to make a specific difference with that approach. And for the last 20 years, I've been working on flow diversion devices in aneurysms as the grounding problem. And recently, I got an ERC advanced grant for actually working on TAVI devices, or transcatheter aortic valve implantations, which is a prosthetic valve in the heart. And I'll show some examples of the things that I told you so far, grounded by these two. And this requires collaboration with clinicians, but also with industry and regulators. So let me start a bit with the aneurysm one. So in the aneurysm one, so this is the vasculature in the brain. An aneurysm is a bulge of the vessels that potentially can burst, and you have a hemorrhagic stroke. They like to appear mostly in the bifurcations of the vessels. There is tons of devices to fix them, but historically people started with clips. Clips require craniotomy, so you need to make a hole in the skull and put a clip. And then people try to find ways to go minimally invasive. So all of these techniques, you go through the groin, so it's the Plummer way. So you go through the leg under catheter guidance all the way up to the brain. The only thing you have is fluoroscopy, so X-ray imaging, to know where you are. And so imagine the operator is in the leg, is like passing a cable, effectively, and gets somewhere into the brain in an artery that has maybe 2 millimeters diameter, and deploys a coil. Or if the neck is too broad, it puts first a scaffolding stent and then the coil by getting through the stent. And then people realize that if you do a stent which is a lot less porous, you don't even need a coil because you can reduce the flow. So what are these guys trying to do with all of this? They're trying to slow down the flow, because we know that if you slow down the flow, you promote coagulation, right? That's what you're trying to produce. And these are just different ways to get to that effect that you want. From the drastic one of putting a clip and taking the aneurysm out, to these other ones which are less invasive but more indirect as well. So we're going to pick one device, one of this family, a flow diverter device, and I will give you an example of coiling later, which is a device from Medtronic, and it's called PED, or pipeline endovascular device. This device was approved by FDA in 2011, and it was approved after loads of small studies. These studies look all big, but actually there are 100 patients, 50 patients, 10 patients, you know. There was one study which is what is called a pivotal trial. This is the study after which you are allowed to go and sell. Okay? So this pivotal trial took 8 years from design to publication, 6 years of recruitment, 108 patients, that's all the evidence. And then we're going to be putting it to everyone in the population. That's what your colleague was saying before. Just to give you an idea, a hospital here in Manchester can put 100 of them a year. Yeah? And then this probably costs somewhere between 30 and 40 million. And that's one trial. And if you know a bit about trials, you know that you need to define a question, you need to collect the data, and then if you change the question, the data is no longer valid, and you need to start all over. So what would look like an In-Silico trial? An In-Silico trial will look something like this. We have the device, and I'm not going to talk a lot about how we model it, but you need to model the device itself. And then imagine that you have access through regional electronic health records and regional PAC systems to all of the imaging of a region. So Greater Manchester, 3 million people. And of course, not everybody will have an angiogram of the brain, but say that, I don't know, 0.5% have that. 1 to 3 million people is quite a lot of people. Definitely 1 or 2 orders of magnitude bigger than you will do the trials today. And then for each of those guys, we're going to throw some image segmentation algorithms that are able, mostly used in deep learning, that will build the 3-dimensional models for each of them. So each of these guys is a copy of the anatomy of a real patient somewhere. Okay? And for each of those, we're going to virtually implant the stent. So you probably see the stent here, is less visible here, but it's there as well. Then you're going to do a fluid dynamic simulation with finite elements before you put the stent and after you put the stent. And then you do the ratio between how much of the blood flow that used to go through that aneurysm is removed after you put the aneurysm. And this is this number, this readout here. We call it reduction in maximum time average velocity at the neck. This is what we call a surrogate endpoint. we would love to be able to predict death, but we don't know how to do that. So what we know is that if that parameter is not high enough, you won't clot the aneurysm, you're essentially not solving the problem. And you can see already that these two patients, the same device, they have different performance. Now imagine that you have wearable sensors, or you have some way to get measurements from the physiology and the lifestyle of the patients. We can create Gaussian process models, and we'll talk a bit about them later, where we model the average patient and the variability across all of the patients, and also because we did the 48 hours Holter to do that, we know what is the average in rest and the average in stress. So you can have rest and stress conditions, and therefore if I take the first patient and the same device in both, so nothing has changed, we're going to change what is called the boundary conditions that represent rest and exercise, and I'll explain later how you can do that. Just changing from rest to exercise, you change the performance of the device. Okay? And then, of course, you can put two competing devices, or you can put one of these devices that we are trying to optimize the strut density, for instance. And for each of these two devices, everything else being fixed, we can look at the difference in performance. Okay? So this is like a multidimensional virtual experimentation space, where every node here is a virtual experiment that combines a certain anatomy, a certain physiology, and a certain device design. And clearly this is not one parameter, it's a family of parameters. And probably I'm missing dimensions here. But you get the idea, right? And what our colleague was saying in the previous talk is that the problem is that this looks like a very sparse space in reality, apparently, in pharma, right? Here, in medical devices, it's also very sparse. But if we can simulate, we can densify it as much as we want, right? If we have ways to create virtual populations that spread all this space appropriately. Where do we get this? Well, we get this from regional data sets, from the device specs from the manufacturer, from population data, and there are data sets coming increasingly that allows you to do this. How does the pipeline look like? And this is what I was asking also the previous colleague is, are your pipelines mechanistic or data-driven? So you'll see here, most of them are actually quite mechanistic. So the data-driven part is essentially how we build geometry, because we don't have equations that tells you the geometry and the variability across patients, or at least not in a mechanistic way. So from the images, we need to create three-dimensional models, clean them up, do tetrahedral meshes, well, surface meshes, tetrahedral meshes. Then we need to develop models for the devices themselves, deploy them, models for the boundary conditions, models for the physics, in this case the Navier-Stokes equations, and sometimes we solve as well equations of the reaction diffusion equations for transport equations of a species in the case of biochemistry of clotting, or of contrast agents if we're trying to do virtual angiograms for validation purposes. So we can look at what the flow looks like after the treatment, and then we also have biochemistry that is coupled to the physics that gives you some maps that I'll explain later that relate to the quality of the clot. Okay? And these are just some of the papers. There's probably about 50 journals behind this pipeline, 50 journal papers that show every single thing that we develop, the validation of those, and also in some cases, things that we don't use, but we have the paper that explains us why we shouldn't be using a certain parameter. Now, you could say, well, how fast is to do this? Well, I'll show you later a trial where it took us probably 3 months to run the trial against the 8 years from a human trial. The part where I'm cheating you is that I'm not telling you that it takes 8 years to develop the models, perhaps. Now, the advantage is if I develop the models, I can use it as many times as I want. If you develop 8 years of data collection for a trial, the next trial you need to start all over. So there is a reusability element that you don't have in all of those. And the other thing which is quite interesting, models are pieces of code, and therefore things that you can put in a GitHub, and therefore it's transparent. You make your assumptions explicit. There is lots of assumptions in a randomized controlled trial that you never quite fully grasp that you are making, right? Because assumption is in the data fundamentally rather than in the code. Okay. The other example is with TAVI. So this is something relatively new that we have. So we have the full data set from the REPRISE 3 randomized controlled trial. This data set cost about 80 million to produce, the data set of the trial alone. And this is a device that sadly was taken out of the market in 2020 because it had problems with the delivery system. The cost for the company of the recall was between 200 and 300 million. Okay? Now, we're going to take that failed device with all the data as to generate twins of each of those subjects and using the randomized control trial as gold standard for our predictions to say, can we predict at the individual level the same outcomes that they got, and can we also predict the group response at the trial level? Okay? Now, you may say, why are you spending, I don't know, so much time into one device? Because actually it's not just one device. This device, the Lotus device, from Boston Scientific, actually there is a whole zoo of them from different manufacturers. And each of them went through similar processes of cost, years, and so on. So if I could show that we can reproduce the findings so that we could create a trust, which is all what we are trying to do here, the evidence of the trust, to regulators and industry, that next time that they need to do a trial, first they should invest $1 million or $2 million, do a virtual one, get reassured themselves that it is worth going to the next level, and only then go and run the trial. This is helping to de-risk patients, but also to de-risk their investment as well. Okay? So what we are trying to do here, we are trying to go from each of the data sets we get to three-dimensional models of the heart, the aorta with a whole valve, and you know, it's again building pipelines about how you go from the anatomy to then the physics, the deployment of the frame of the device. And one of the problems of this device is that, so there are two families of devices in the market. Some of them have the problem that they have leakage of blood around the frame of the device. And some of them have the problem that they deploy too strongly against the aorta, and if you know a bit about anatomy, you know that all the conduction system, the peripheral conduction system of the heart that communicates the atria with the ventricles and propagates electrical conduction go through that part. And if you overstretch the aorta, what you're doing is you're breaking the Purkinje system effectively. And about 30% of those patients, a week later, they need to go back to the hospital to put a pacemaker. Okay? Because you mess up with it. So it's an excellent optimization. Again, another problem for machine learning, optimization problem, where you have a two objective function. Don't open too much the frame so that you don't mess up with the conduction system. Don't do it too loose because otherwise you get perivalvular leakage. Right? Okay, let's go now to some proper examples in more details based on those two large families of problems that I'm working on. This one, I'm going to show you an end-to-end in-silico trial, and I'm going to exemplify a few things, how we reenact how we do an in-silico reenactment of a real trial, how you incorporate physics, biology, and physiology into one of these trials, and also how a model can start producing emerging behaviors that are not actually coded in the model. And this is one of the things that FDA uses as one of the eight credibility factors for a model. So if a model starts predicting things that are not coded and they're known, that's the good stuff. And if it starts predicting things you didn't even know, that then get confirmed, even better, right? So again, the device, how this device works. So you put this flow diversion device from Medtronic into the vessel. You put it this way. This reduces the blood flow in the aneurysm, and it produces thrombus. This thrombus essentially eliminates the aneurysm from the circulation, and everybody's happy. Now, you cannot put a graft like you do in the aorta. You need to use these flow diversion devices that still have porosity through them, because otherwise you mess up with the perforator arteries, which are the ones that feed the brain themselves. And you sort out an hemorrhagic stroke, but you produce an ischemic stroke with that. Now, this device, the one we talked was introduced in 2011, is this one in orange, and you see how the market share of this device very quickly pick up the whole market. So you get one of these new innovations, and you better get it right, because if it's reasonably good, everybody will start using it. So if you have anything that can go wrong, will start going wrong very quickly, and you will see it. Look at the list of all the different products. I did it just to fit the page, but not necessarily that you could read what is all there. Each of these is a product from a particular company. These are coils, these are stents, these are flow diverters, these are other devices like glues and other stuff there is. These are balloons. It's a zoo. Each of them, think about a trial, 6 to 8 years, 20 million, 30 million. Half of them will get it wrong. It's just hopeless. So this is the question we wanted to answer. Can an in-silico trial replicate and hopefully expand conventional clinical trials? So we published about 2 years ago a paper that we are super proud of in Nature Communications, where we demonstrated, as the title says, that that's the case. And I'm going to talk a bit about through that. So our in-silico trial is called FDPass. And again, I'm not trying here to go over all the details, but I want to highlight that you design it as any other trial with, certain errors, certain number required of subjects, as a single arm, whatever. And then this is how we're going to build all of these pages. So we first start with a library of models. This is something that when we did this work, we didn't have generative models of the vasculature. And I'll talk about later the work we're trying to do in this direction. So the best I could do is my generative model was an enumeration of cases. So I have a library of cases. And I iterated over them. So there was 82. And because I didn't have the data from the actual trial, what I had to do is compare the characteristics of the data that I could get my hands on with the characteristics of POOFs, which is the pivotal trial, Premier, which is the trial that followed this one. You see that the aneurysms here are smaller than the aneurysms here. These are 18 millimeters on average. These are 5 millimeters on average. So the first trial went for the big ones because they are the ones at risk. The second trial, they got excited and they said, well, if we can treat the big ones, can we also treat the small ones, rather than wait until they become big? But you wouldn't do that before you prove the device works, because these people are less at risk than the ones, these people are less at risk than these ones, right? These are unruptured and these are ruptured aneurysms. And the Aspire is what is called a registry. So a registry is, once you get your trials approved, you're allowed to go to market, but you need to keep a tab about what's going on. And every time there is an adverse effect or a problem, you need to keep controlling that. And that is what is called a registry. The list is lighter in the amount of data and in the quality of the data in a sense, but it's more extensive and normally is a follow-up. So you get signals of risk. If something starts going wrong in this group, you need to stop, you need to analyze, and that's another potential application of in-silico trials. Say that you start seeing that all the men from 45 years old that are a smoker, all of them tend to have complications. Well, you can do a zoom through simulation that you try to look at what's going on with that subgroup before you allow that to accrue new adverse effects. Anyway, so we demonstrated working with the trialists that this was okay to use these data in lieu of those ones. Now we need to put physiology to those. And because we didn't have, and we'll come back to that this is life, most of the times you don't have all the data you want. So you have anatomy, but you don't have physiology. You have physiology, but you don't have imaging, right? Because there are different types of experiments. And in the NHS, nobody goes and says, what do the machine learning people need? Let's collect everything for them. The way they work is, what does the patient need? And if the patient only needs imaging, they will not do anything else. Or if they only need an ECG, they will not do an MR scan. So the data is collected on the basis of needs, as opposed to the basis of opportunities of what I can do with it. So we have developed methods that allows us to piece together the data so we can create virtual patients nonetheless. And we don't call them digital twins because they're not twins technically. We call them digital chimeras, as I'll comment in a minute. So say this operation that I do it as a nice motion, but this is what I call blending of these two arms. So this modality 1, modality 2, and we do some sort of a statistical enrichment and blending so that we have, instead of one axis and another axis, or one axis and the other axis, we create an outer product of all of those. So we create a space, right? So every dot here is one of these virtual patients, and then for each of those guys, we need to develop, deploy our algorithms for putting the devices. We then need to solve the physics. So here, these is particle simulations where we put a bunch of particles downstream and we let them drift with the blood flow. And what you see is that there is more residence time within the aneurysm after putting the stent than before, because that's precisely what the stent does, it slows down the flow, right? Then we can also look at what happens if you have a patient that is normotensive or if a patient is hypertensive by controlling the boundary conditions. And you'll see why we do that in a minute. And you see that the average mean velocity reduction rate goes down a bit more in the hypertensive sorry, less in the hypertensive than in the normotensive. And then you need to solve the coupling of the Navier-Stokes equations with the diffusion, the reaction diffusion equations of the biochemistry of clotting. Now, do you see something weird there happening? You have an arrow, so you can't just miss it. What is happening there? We're trying to fix a hemorrhagic stroke, right? We're trying to remove the aneurysm from the circulation, because if this thing keeps there, the pressure will eventually burst it. And in doing so, we found that what are we doing? Is there any neurologist on the? So what we are finding is that in this particular case, same geometry, same device, everything is the same. The only thing that is different, this is normotensive, this is hypertensive. It turns out the blood clot comes into the vessel. So it starts blocking the side branch that, remember, feeds the brain. So that's going to generate an ischemic stroke that you don't want. Now, you may say, well, is this anecdotal evidence? You go to the literature, and there is no trial that shows them, but there's a bunch of case studies here and there that says, don't put the stents when there is a side branch. We really don't know why. We don't do that because the few that we did is always a problem. So we then realized that from the 82 examples that we had, cases, 27 had a side branch. And this phenomenon happened in every single one of them. So if you have a side branch and there is hypertension, you will get a clot in the vessel. So this is important because this is a failure mode of the device that you need to know that is likely to happen. What can you do? Well, maybe you can improve the design. Maybe you need to put a hypertensive treatment to this patient before it goes to be treated. So the hypertension comes down to as close as possible to a normotensive phenotype. And that is an issue. If you want to do a drug, put drug to these patients and put a device, this is a combination therapy, is a combinatorial test, because now you have a trial where you need to combine for different types of devices or different positions or different versions of the treatment and different amounts of dosage of the drug, how those two things interact. And I wish you luck to go and do a trial for that, because this multiplies enormously the complexity and the size of the trial, and what drug you use then. And then, so these are things you can do in modeling that are very difficult to do otherwise. And you can get to the root causes. So you see, this is what I was saying. It may be that a technical endpoint is there a side branch that is, whose patency has been compromised? This is probably more interesting than knowing who of them died and who didn't. Because if you only know that they died, it's like you know the car is not working, but you don't understand whether it's the battery, the petrol, or what is the root cause that is leading to that. Okay, and then once you have all of these virtual images, because simulations are virtual images, you represent the outputs pretty much like you do with any trial. It's just that you read them from simulations rather than from real data. So you do all of this, and these are the results. So the big headline results. So if you do the in-silico trial of this device with all of the patients being normotensive, this is the percentage of those patients that respond positively. If all of them per counterpart are hypertensive, these go down substantially. If you do the average of these two, it's about 75%, which is pretty much what you get with all of the other trials. None of these trials control for hypertension, because there's 100 patients, they don't have enough numbers to actually do that. We did a lot more than that, but I'll leave you with that headline. And it's interesting that some of the other things and the findings we do, if you do relative risk, odds ratio and this sort of stuff, the bigger the odds ratio is, the more consistent is with the literature that says that there is a very clear, a very clear direction for the effect. While some of the cases we studied that have odds ratios that are much smaller, the literature about the influence of hypertension is a bit split. Okay? Okay. By the way, how do we come up with these ideas about the particular study? Well, working with clinicians. That's fundamental. I don't need to tell you. And it's not like, to me, many people from our community, and I feel that I'm part of this community as you are, We think that clinicians are important to give us data. I believe that the most important thing a clinician can give me is hypothesis. The data normally follows. If a clinician cannot give you hypothesis, he's unlikely to have the data, and more importantly, you will have data that you don't know what to do with it, even if you have some data. So work with them to answer questions together rather than, and the data will follow. So in this case, it's a colleague who is an honorary professor here in Manchester, but he's clinician in Leeds. And he was the one who helped us to develop a new study that we're doing at the moment, where he's using his international network to collect cases so we can answer those. Second case study, same device, off-label use. So off-label use is when a device that is, say, approved for adults is used off-label, used for children, for instance, right? Or is used for women who are pregnant when that was an exclusion criteria in your trial. Yeah? So this should be saying here, cerebral, not communicating artery. So the posterior cerebral artery is a type of artery that in some patients, in some people, all of them being normal, so some of you here, some of us, will have this segment in the brain, some of us will not have this segment. And some of us will have this segment with hypoplasia, which means is smaller in diameter. So it turns out that this posterior cerebral artery has been, sorry, this is correct. This is posterior communicating. I keep confusing the two. So the fact is we are trying to look at what happens with stents put in this area, which is the posterior communicating, depending on whether what happens with the posterior cerebral artery, which is in this location. So this device, the PED device that I talked before, it was explicitly excluded. Sorry, the aneurysms in the posterior communicating artery are specifically excluded. It's an exclusion criteria for the trial that I showed before. Therefore, the device has not been approved for those locations. And there are some people, and it's about 25% of the aneurysms, right? And there are some variants, which is this fetal variant, and the which is the one that you have here when there is this effect. And what is interesting is that these fetal and non-fetal are heavily dependent on the ethnicity of the patients. So the non-fetal variant is more prevalent in Black and in Hispanic communities. So the question is, how does this device perform in those areas, and could we expand the prescription of the device to those areas as well? So again, I'm not going to go through all of the details, but there is a proper design of each of those trials and a pipeline that you need to lay out. Many of these components are from the trial that I talked before. That's where you have the reusability of components as well that I was talking about. And essentially we were asking a primary and a secondary hypothesis. The primary hypothesis is there a maintained posterior communicating artery patency post-flow diversion in patients, and how that affects the success in patients with fetal posterior circulation. And the secondary analysis is understand the influence of the P, and aneurysm morphology on treatment efficacy. So for the secondary analysis, we show that the measure of flow reduction that we use as an endpoint is flat, irrespective of many aneurysm anatomy sort of parameters, aneurysm diameter, neck diameter, aspect ratio, non-sphericity. So it doesn't matter, basically. It behaves the same in all of them. And then, again, you need to reproduce the flow conditions of the fetal and the non-fetal components. So this is what is called the fetal variant, which is the one where you completely lack that posterior cerebral artery, and the non-fetal one, which is this one, which is where you have the whole circle of Willis properly. Okay? And if you do that and you put the conditions, you demonstrate, you answer the primary hypothesis, which is the flow diverter treatment efficacy is effectively reduced in patients with fetal posterior communication. So actually there is a problem with those devices. That can lead the manufacturers to rethink their design and also to realize that they have a problem of equity in the way that they treat patients, because certain groups are going to have less solutions from their firm than others. So if they want to be more, promoting EDI in a sense with their products, they will have to put some R&D to think about that problem. And the third example, I'm going to change now the device, and I'm going to look at what is the influence of the operator, because these devices need to be put by a human being. So is it, when there is a human in the loop, does it have an effect or not? So let me explain you first what these devices look like. So these are coils. These historically are previous to the flow diversion devices, and after the first devices after the clipping. They have shape memory, so they go through some process of materials conforming that they can be put within the sheath of the guide wire and the sheath of the device themselves, and when they are within that, they're straight, so you can manipulate and bring them to the place, and then you start taking out the sheath, which is like the external cover of the device, and they start curling around with the memory that you pre-set in those materials, okay? These are usually nitinol type of materials. And some of them also have some drugs on top of them and some gels that are prothrombotic. So there's all kind of issues, different people have different skills, different outcomes. And look, another zoo of devices with all kind of different things from different manufacturers. One key parameter is what we call the packing density. So if this is the aneurysm, and say it has volume 1, what is the volume of metal you're putting within that unitary volume? And the packing rate, the higher it is, the more metal you're putting, and ideally, if you were able to fill it fully, you will have a packing rate of 1. This is impossible. Packing rates of 0.3, 0.35 is almost the most that people can achieve. And again, this is a problem it's a mathematical problem of how much you can compact certain shapes within others, right? Now, this is how they look like when you do a surgical specimen. These are the struts in a histological sector, in a histological section. So how you develop a method to solve the virtual placement of one of those? Well, we developed a number of years ago, back in 2013, a method which is using a minimum cost path and a Dijkstra type of algorithm that essentially what it does, it sort of tries to fill in the space without going through the wall, without self-penetrating, and without doing weird things like 180-degree bendings that are physically impossible. Right? So there is a combination of some heuristics. The second one is how do you validate a model that does all of that? Well, you can model it, you can validate it with rabbit models where we had rabbit models of aneurysms where coils were put, and we know exactly the distribution of the struts. And then what we do is we take our models and we do a virtual histology. We cut the models with a plane, and we look at how those struts are distributed and whether the distribution of the struts in the periphery and in the core and in the neck and towards the end of the aneurysm has the same density in the physical experiment that in the virtual experiment, right? And this is the way I think we will be using animal models in the future. Rather than to do that in the extent we use at the moment, we will use them to calibrate and validate models that then will be used to do inferences on subjects. And another way in which you can validate with these models that we also did is by, if you have a 3D-dimensional model of the vessel, and you solve the Navier-Stokes equations for flow, and then you solve the transport equations for the contrast media, for the volutes of contrast media. So this is iodinated contrast that you put through, you flush through the vessel, and then these iodinated contrast agents in X-ray, they're dark, they're black. This blackness comes from the iodine. So what you can do is solve the transport equations for the contrast, and then do a simulated X-ray by doing ray casting and accumulating density, and look at whether the virtual angiogram and the real angiogram, how they compare to each other. So what I'm showing you here is, I think the CFD is the dotted line, and the continuous line is the actual time dilution curve. So you put a point in that black in that position in the vessel, and you look at how the intensity goes up and down. That's the density of contrast, right? So these wash-in and wash-out curves, you can compare them with the virtual wash-in and wash-out curves of your CFD. And if the model is good, you should basically, put in one on top of the other, and you can see the results are pretty much spot on, with the exception of some areas where we have the inlets fundamentally, which is where there is a lot of boundary effects. Now, okay, so say that you believe the models of coiling. Now, what can you do with this? Because let me try to remind you what I'm trying to do. I'm trying to predict where the coils will land once some operator goes and puts them. This is crazy, because this is like an stochastic field, right? So they're going to land anywhere, statistically. There's no way to say I'm going to put it in this position. Remember, we are entering through the groin, and we are pulling and pushing and doing torsion and trying to get into the the only thing you're trying is to fill the aneurysm. So let me convince you that this has some sense. So what is this table here? So the first column, the second column, and the third column, the only difference is this 20, 25, 32. This is the packing rate that I was talking before. And what is configuration A, B, and C? This is, if this was an stochastic field, this is realizations of that stochastic field. What do I mean by this? Say that I'm going to work at 20%. So I put coils, enough coils, and each color is a different coil. I know the diameter, I know the length, so I can calculate the volume. So I put enough coils to fill this aneurysm to a 20%. And they land wherever. Then I take them out, which I couldn't do in a real patient, and I put them again, and they're going to land in a different area because there is some element of stochastic initialization when we put the first coil. But I put again enough to 20%. That's configuration 2. Then I take them out, and I put them again. And I can do that 50 times if you want. So what we demonstrate, so you follow me, right? So what we are doing is we are filling the aneurysm to 20% in different ways. Yeah? The only thing that is constant is the filling rate. And we can do to 20% constant filling rate, to 25%, to 32%, to whatever percentage you tell me. Now, what we observe by doing this virtual experiment is that if you plot the average flow reduction rate, how much we slow down the flow, against the density, or the average wall shear stress of the wall against the density, all of these quantities more or less go to an asymptotic steady state after a certain amount of packing. So that there is a point where if you keep putting coils, the only thing you're doing is wasting money at the rate of 1,000 pounds per coil. You're overpressurizing the aneurysm with the risk of potentially bursting it on the intervention, and you're not making any difference to the patient. If you put below that number, so if you're in this regime, then what happens, only God knows. Because it depends on how expert you are, how good you are, how much exactly you put. So it's very operator dependent, the outcome. So this is interesting, right? Because now we know through this model what is the optimal packing rate for a particular individual that gives you the best use of money and the best possible outcome for each of them. Now, 10 years before, so this paper was published in 2013, 2002, there was a paper on real subjects where they show that the embolization volume, the packing rate, is higher in patients that are stable, meaning they don't have problems down the line, compared to the unstable aneurysms that reopen. And also that if the aneurysm was small, the packing rate was different than if the aneurysm was large. And in fact, if the neck was small or large, also made a difference. Of course, because this is a 3-vein clustering of what effectively is a patient-specific stratification, right? And yes, so what I want you to get from this is an in-silico trial is not just a way to generate the optimal design, but also can help you to develop a stratification chart so that you use the best technology for the particular patient that you have under the table. I wanted to show quickly, and I'm going to run through this, some of the methods under the hood. So this is kind of a pipeline. So the first thing we need to do, if you are from, how many of you are working in computer vision or in imaging? Can you raise your hand? Okay. So you will know what we mean when we say segmentation, right? So first problem, that I think all of us who came from computer vision, we will probably start and die in our career solving this problem. I don't think it has a solution, but there are smart things you can do for particular problems. It doesn't have a solution in general, I mean. So what data we use? Well, you can use three-dimensional rotational angiography. So this is the type of imaging you do during an intervention. This is nice because it's isotropic, it's high resolution, but you need to put a catheter on the patient. So it's intra-interventional. Or MR, which is not so nice, bigger resolution, lower resolution, anisotropic voxels. But the nice thing of this is it covers a full circle of Willis because you don't need contrast injection, and you can do it non-invasively, and you can do it before you even put the patient on the table. Why this is difficult? Well, because, well, ask someone who did segmentation, and they will tell you 50 reasons why it's complicated. But in this particular problem, these vessels, we are talking about vessels that are 2-3 voxels across the diameter. So there is problems of kissing vessels, two vessels that run so close to each other that they blend, missing vessels, vessels that actually should be connecting two other parts that are lacking because they're almost at resolution level. And we used to solve this many years ago. Probably many of you will not even know what I'm going to be talking about, level sets, if that means something to anybody. And it used to take 6 weeks to do 82 cases. It was art more than science and a lot of laborious operation dependency. We now can do all of this in 30 minutes using deep learning techniques. So, as I said, there are moments where data-driven phenomenological models are the right thing to do. Segmentation, I would say, is one of them. But it's not just segmenting, my friends. And this is the problem. You need to have a large phenotyping capability because segmenting is only the beginning. I also want the center lines. I want to detect I need to segment the aneurysm with respect to the rest of the vessel. I need to know where the neck is. I need to be able to release a device. I need to be able to do then set boundary conditions automatically, which means I need to know which particular position of the circle of Willis these and these are. And all of that, high throughput, quality control, lots of AI there as well. We developed lots of algorithms. I'm not going to get to that, but it's a heck of validation and cross-checking and demonstrating the accuracy that usually goes under the radar when I present the rest of the work. We also developed techniques for automatic labeling of the vessels. So this is the full circle of Willis. And if my aneurysm is in this position and I could know that this is the anterior communicating artery somehow, without someone having to pinpoint that, I can set boundary conditions automatically because I can have an atlas of blood flow where I know the blood flow in every possible branch, right? But doing a labeling is a complicated problem because this is a graph. You can represent this as a graph. And the interesting thing is there is about 8 variants of the circle of Willis, all of which are actually are physiologically correct. So it's a problem of graph matching with another graph, where actually it's not with one, it's 8 graphs, so that you can identify, is this graph that I derive from an image matching to any of my 8 templates? And once I know to which it matches, I can then infer the labels from the atlas as well. And sometimes the graph that I derive from the image is incomplete because the image is not perfect, because I'm missing vessels. So it's a graph matching under incomplete data and a lot of uncertainty, right? And then you end up with putting colors to each of those labels, which correspond the name of whatever vessel it is. And then once you have that, you can have a lookup table that says if you're in the green area, this is the anterior communicating artery, you need to use these boundary conditions for a patient of this sex and age and body mass index like the one you're considering. The other one is about how you set boundary conditions. So remember, we're trying to solve the flow in a little piece of the brain. This little piece of the brain is connected to the rest of the brain and it's connected to the rest of the circulation. You cannot do a 3D flow simulation of the whole body because that takes forever and it's irrelevant for the problem you have. So what you need to do is multi-scale coupling, where you have one-dimensional or even zero-dimensional models of the circulatory system, where you model everything else that you're not interested in, and the particular part that you're interested in, you replace the particular, I think I have a picture later on, you replace a particular segment by your 3-dimensional detail model. How you get these measurements, you get them with wearables, you get them with ICU data, and again, you need to model uncertainty. So how we do that, So for instance, we developed this sort of, by the way, this is the model I was looking for. So you have this model of the whole body, the circle of Willis, you do it with the 1D model, the particular area of the aneurysm, you do it with the 3D, and the rest of the body you do with 0D models. So these are electrical equivalents of blood flow. Now, in all of these models that are 1 and 0 dimensional, you can put uncertainty. And these are the models that we model with Gaussian processes, where, again, without getting to the super detail, but we can get things like this, where for a particular height of the patient, and whether it's male or female, and you can get what is the scaling that we need to do to the waveform that we measure to sort of customize to that particular patient. Okay? Why this is useful? Because then you can generate chimeric or counterfactual scenarios. I can say, what happens if this patient has different levels of stress? Or what happens if, for a particular patient, I don't actually know what the flow is, and I want to look at the sensitivity due to the lack of knowledge that I have on that variable. How much this matters to my ultimate result on the trial. I also mentioned that we do clotting models. These are, again, all these equations where we model exchange of species, and we also develop some metrics that tells us the quality of the blood. So it turns out that there is a type of blood clot which is called red clot that is poor in platelets and fibrin, and this is like a gels that can redilute, while the platelet and fibrin rich clot or white clot, this is like a fiber reinforced material that is actually quite stable. And we can predict what sort of clot will happen for a particular anatomy and a particular setup. How we validate that? Well, there are experiments out there that show for 2 different experimental setups with different struts what the size of the clot will be, and we sort of demonstrate that our predictions are more or less in line with those experiments. So experiments are important. I'm not trying to sell that models are the only thing, but they're important to develop and validate the models, and then you use the models to do the sophisticated interaction bit. I want to show one last thing, and then I'll skip the rest. This is the point about, are you a theoretician or are you an applied? I don't know what I am. But what I know is that to solve real problems, you need to have a heck of a complicated technique sometimes, and you need to develop one. So the ERC that I got is in informatics, so it's not in applications, it's in the computer science side. And fundamentally, what we're trying to do is develop a number of fundamental components of machine learning that will enable to do a lot of the things that I mentioned before. So one is how we learn over hybrid, complex, and multi-scale representations. The other one is how we do joint learning over data sets of data sets. And the other one is how we use multi-scale physics informed over graph representations. And I'm going to skip the whole thing because it's just too complicated to go over this. But to show some of the virtual populations bit. So what people used to do to build virtual populations in the past was these sort of potatoid shapes that was supposedly some sort of thorax for people using it in MR and physics. Then someone used the virtual, the visible, what was it? Anyway, there was this guy who was apparently in prison and sentenced to death in Germany, and then he gifted his body to science after he was executed, and then the guy was imaged to death basically with a lot of high resolution, and they created these 3-dimensional models that many people use, n equal 1, okay? Then someone said, well, this is too narrow, right? It's just n equal 1. So they started to do that with, by then with MR, which was less invasive, with a number of people, and they created these virtual families, which are effectively imaging and someone who has spent endless hours contouring, right? So what we are trying to do is to scale that up. So if this was a digital twin, and you have a population, say from a trial, where you give treatment brown to one's arm and treatment black to the other one, doing a digital twins means that for each of these guys, you generate a digital replica, yeah? And therefore, your cube of experiments is super sparse. So what we are developing, and we've done some publications and get some patents on that, is this concept of virtual chimeras, which effectively what we do is we do generative models at source, and then we do blending statistically from them, so that you can synthesize much more than you had in the beginning, and therefore cover much better the whole population. The idea here, and it solves a number of problems. It solves the problem of GDPR as well, because you make models at source. So by the time you blend, there is no way you can go all the way back, particularly if you put things like differential privacy in your generative models. There is no way you will even know whether a particular subject was part of your training set. But also, it solves other issues, which is the one that I was saying earlier about how data is collected. I would love to have anatomy, invasive measurements, tissue characterization, wearables. If I start a conversation like this with the clinicians, I lose them in 5 minutes because it's impossible to collect that. You won't collect that data. But what is possible is to collect lots of data on imaging, lots of data on, say, wearables, lots of data on other things, and then do an observation. Is that if you are, say, in Greater Manchester, and you have lots of people from Greater Manchester that have imaging, and lots of people from Greater Manchester that have wearables, there are two cohorts of the same population. So what you're observing is marginal probabilities of a multidimensional probability function that you are trying to infer, right? So it's like you have projections of your probability distribution through some sort of vistas of modalities. And the question is, can I recover the multimodality reality from those vistas that are not paired, but are coming from the same population? So that's what we call a virtual chimera. It's solving that problem. Because these guys that we create, these guys here, they're none of these guys at the beginning. They are like one of them. They're statistically indistinguishable because they are like drawn from the same distribution, and they have all the modalities within them. So you could see the problem of virtual populations as a representation learning problem, where you have a reality, the heart, the full heart. And by the way, you can generalize this idea of vistas to multiple things. So one could be different. That's what we talk about, data sets of data sets. It could also be that you have, from a lot of people, 3D CT, but you don't have 3D echo from them. But you have another group of people from the same population from where you have lots of echo data, from where you can see biodynamics, for instance. Can you blend it all together? So this is basically the idea. You have a structure missingness by sampling, where you only see certain vistas from different subgroups. We put that into some smart network that effectively synthesizes the whole. And then we can synthesize virtual populations of the whole thing. Okay? So we published that a few weeks ago, came online in Transactions on Neural Networks and Learning Systems. And this is only the very first version that we have. There's a second one we're working at the moment that will allow to do conditional synthesis. So you can actually put inclusion and exclusion criteria, right? So what we are trying to do is, I'm going to skip all of this and show you some examples. This is basically all the experiments we did to demonstrate that, because we are using UK Biobank to do this, and the beauty of UK Biobank is that you can have a population from where you have all the vistas. So you can synthetically eliminate, say, the ventricles from a subset, from another subset, the atria, then blend them together, synthesize the part that was missing, and compare against the ground truths that you did have from all of those subjects. And what we did all the experiments to demonstrate that by doing different things, you have good generalization and good specificity properties. So I'm not going to waste your time by going through all of those. If you trust me that we did all the experiments. But I want to show you this one. This is the second generation of what we are doing, which is with, someone was asking me earlier in the back, do you do things on diffusion models? Yes. So these are the sort of normalizing flows where we actually we have encoders and decoders, but we also can synthesize, we can put a conditional branch that, which is this one here, that puts the demographics. So I'll leave this. So we train, and then we synthesize, and we say, look, give me only women. Give me only women from 45 to 65 years old. Give me people who have hypertension. And then you can synthesize virtual populations for that subgroup, and then you can put devices. And these are all the experiments to compare the virtual population with the real population. So how we have the real and the virtual, because, again, in UK Biobank, we can actually give a verification set outside and compare our predictions, in-class predictions, and we are at the moment working about doing extrapolation outside of the class. So if we want to go to age groups that are not in the study. And our dream is to be able to do this with data at the scale of a region, because then you're not doing that with 60, 80,000 cases like UK Biobank, which is great, but you can do that with a 3 million population. And the likelihood that you don't generalize well goes low and low as you grow. And then this is our dream for building a virtual CRO, or virtual contract research organization, if you're into the jargon, where we can specify the characteristics. So all of these characteristics, age, haze, weight, these are all the metadata from UK Biobank for each of those images that we have. So we can put them as conditionals, as ranges, or as values that we fix. And then you synthesize subjects that are all meeting all of those inclusion and exclusion criteria. And this is super powerful. Okay? Anyway, I think we can do things like exploring virtual patients scenarios where they gain weight or when they get older. And we can compare them for a number of parameters. And we are working in developing metrics and theory for how do we say that these models are representing the real population. And there is work going on with colleagues in computer science that are looking at certification of generative models, because if we have ways to certify these models, this is what a regulator wants down the line. Remember that slide that I showed from MHRA showed me that your virtual populations are like the real ones. Imagine that we could demonstrate and provide guarantees that our model will satisfy those. And then the final thing is you can also look at synthesis, so virtual populations as a data synthesis problem. Let me show you another example. So remember, we are trying to do also the aneurysm thing, but because we didn't know how to do the synthesis at the point we did that study, we were working with that enumeration. What we are trying to do now is to build generative models of vascular trees. Now, this is a heck of more complicated than with hearts because the topology changes. So you need to have a generative model that doesn't work like the one we developed for the heart initially, but it works, for instance, with diffusion models that allows you to have that flexibility. And this is something we have done some initial steps. But the other thing is the problem with aneurysms is that you need, in order to have images for building larger scale models, you need to have an angiographic examination of the brain. And usually that requires injection of contrast media and is an invasive technique. So UK Biobank doesn't have that. UK Biobank gives you T1, T2 and protodensity mapping of the brain, but no angiographic scan. So what we are developing is a technique so that starting from these images, which are non-angiographic, can we synthesize the virtual magnetic resonance angiographies. And I'll show you just this image to try to explain this. So what I'm showing there is a maximum intensity projection, is lab 1, where one of these two guys is a real MRA. The other one is a synthetic MRA that was synthesized only with a T1, T2, and proton density from the same subject. So it has the same anatomy, but it's synthetic. And the question is, which of these two is the fake one, and which one is the correct one? And it's not easy, I can tell you. So this is a synthetic one, okay? But we did a lot of experiments about how to do this properly. This is a sophisticated network that is multi-scale because you have vessels which are very tiny, vessels that are very big, so you need to be able to synthesize detail and synthesize coarse anatomy. We also demonstrated that if you segment the ground truths or if you segment the synthetic image, you get essentially the same anatomy, essentially the same fluid dynamics, and the same flow patterns. Yeah? Okay, I'm done. So hopefully what you learn from all of this is that we need to change the way we do product innovation and the regulatory process, that modeling and simulation, of which machine learning and deep learning is just one way of modeling, is essential for that. That this is not just, we haven't gone into that a lot, but it's not just technical. In fact, we have a lot of the tools, but it's also a cultural shift in the regulators, the way they think. That while this is already happening, it needs a lot of cross-regulators engagement and people that think out of the box, that they don't see only the world through the lens of I don't know, whatever you do in machine learning. Yeah, and that it's important, and we're doing that as well, to work with patients, because we spent a few hours 2 or 3 weeks ago in our first public patient engagement in this area, and when I was explaining all of this, the clinicians said, why are we still doing it the way we're doing it? Because for them it was like, that sounds all reasonable, right? You don't want to believe how much regulators are worried about what patients think about all of this. I'm not sure they understand what the patients think about what they're doing at the moment, and how unreasonable it sounds when you explain it to someone that is like us, a layperson, effectively, from the regulatory point of view. So I hope I elicit some enthusiasm from this, and I show you a balance between some of the algorithms that are happening and how we do things, but also why we're doing it. And hopefully a few of you are interested in working in this domain. Thank you.